Okay, sorry about that. So where we left off is ATP provides energy that will perform work for cells. So the cellular work that can occur is mechanical, transport, or chemical, and it's powered by ATP hydrolysis. In the cell, energy from an exergonic hydrolysis of ATP is used to drive endergonic reactions, and overall, the couple reaction is exergonic. Phosphorylation, the transfer of a phosphate group from ATP to another molecule, is typically used to power endergonic reactions. The recipient molecule, a phosphorylated intermediate, is more reactive or less stable with more free energy than the original molecule. Transport and mechanical work in the cell are also nearly always powered by ATP hydrolysis. And ATP hydrolysis causes a change in the protein shape and binding ability. So what type of work can occur? You can get transport, such as moving ions or molecules across by the hydrolysis of ATP. You can get mechanical work, moving things around the cell by hydrolysis of ATP. Enzymes will work by the hydrolysis of ATP. So there's a lot of processes within the cell that will use ATP, harness the energy off of ATP. Now, ATP must be regenerated. Cells need to have a constant supply of energy, just like your bank accounts need to be continuously supplied with funds. ATP is regenerated by the addition of a phosphate group to ADP, which is adenosine diphosphate. Free energy needed to phosphorylate ADP comes from exergonic breakdown reactions or catabolic reactions, such as the breakdown of glucose. The shuttling of inorganic phosphate and energy is called the ATP cycle. It couples energy yielding processes to energy consuming ones so that you have a constant use of ATP, generate ATP, use of ATP, generate ATP. And you can see that here. Now, enzymes will help to speed up metabolic reactions by lowering energy barriers. Spontaneous reactions do not need added energy, but they can be slow, and they could take hundreds or thousands of years to complete. For example, the hydrolysis of sucrose to glucose and fructose is spontaneous. At room temperature, a solution of sucrose in sterile water would sit for years without appreciable hydrolysis. A catalyst is a chemical agent that will speed up a reaction without being consumed by the reaction, meaning the catalyst never becomes part of the reaction. It will never become part of the product. An enzyme is a protein that acts as a catalyst. An enzyme is referred to as a biological catalyst, and the enzyme will speed up specific reactions. For example, adding the enzyme sucrase to the solution at room temperature will catalyze the complete hydrolysis of sucrose within seconds instead of waiting years and years for this to occur naturally. So here we have sucrose, the addition of water, the enzyme breaks it, and you have glucose and fructose. So the active energy barrier, every chemical reaction between molecules involves bond breaking and bond forming. A molecule must be contorted into a highly unstable state before bonds can break and start the reaction. To reach this state, the molecules must absorb energy from its surroundings. The initial energy needed to break the bonds of the reactants is called the activation energy. Heat in the form of thermal energy absorbed from the surroundings often supplies activation energy. Molecules become unstable when enough energy is absorbed to break bonds. This is the transition state. As atoms settle into new, more stable bonds, energy is released to the surroundings. In an exergonic reaction, the formation of new bonds releases more energy than was invested in breaking old bonds. So here we have our reactants, the transition state, and then our products. And in order to get to the transition state, you have to climb that hill of the activation energy. The activation energy provides a barrier that determines the rate of spontaneous reactions. For some reactions, the activation energy is low enough that thermal energy at room temperature is sufficient. Most reactions, however, have a high activation energy and need additional energy, usually heat, to reach the transition state. So how do enzymes speed up reactions? Now, enzymes are defined as lowering the activation energy. Adding heat is not a useful way to speed up reactions in cells because it can cause proteins, which are the major workhorse of cells, to denature, unfold. Heat is also impractical because it would speed up all reactions, not just the ones that are needed. So instead, an organism carries out catalysis, the process by which a catalyst selectively speeds up a reaction without itself being consumed or used by the reaction. An enzyme catalyzes a reaction by lowering the activation energy barrier enough for the reaction to occur at moderate temperatures. An enzyme cannot change free energy. It only speeds up a reaction that would eventually occur anyway. Now, the reactant that the enzyme acts on is called the enzyme's substrate. The enzyme binds its substrate, forming an enzyme-substrate complex. While bound, the catalytic activity of the enzyme converts substrate to a product. Most enzyme names end in ASE or ACE.